My name is John Lorsch, and I'm the director of the National Institute of General Medical Sciences. And I'd like to welcome you to the DeWitt Stetten Jr. lecture. This lecture series honors DeWitt Stetten Jr., who was the third director of NIGMS and who had a strong commitment to basic science, especially research in genetics, cellular and molecular biology, and chemistry. He spent most of his career at NIH serving in 2 institutes and also in the office of the NIH director. He was particularly well known as a co-author of the early editions of the textbook Principles of Biochemistry. In recognition of Dr. Stetten's many contributions, he has the rare distinction of having not 1, but 2 NIH entities named after him. One of them, obviously, is this lecture, and the other is the DeWitt Stetten Jr. Museum of Medical Research, which, if we were in person in the clinical center auditorium, you'd be able to go and see after the lecture, but you'll have to wait until we are all back in person. Trevor Bedford joins a distinguished list of Stetten lecturers. He is currently an associate professor in the Vaccine and Infectious Disease, Public Health Sciences, and Human Biology divisions at the Fred Hutch Cancer Research Center. He's also an affiliate associate professor of epidemiology and genome sciences at the University of Washington. He uses computational methods to study the rapid spread and evolution of viruses, clearly a topic of considerable importance at the moment. His work helps researchers develop successful strategies for monitoring and controlling infectious diseases. He co-developed an open source platform called NextStrain that provides continually updated virus genomic data alongside powerful analytic and visualization tools for researchers. He received a BA in Biological Sciences from the University of Chicago in 2002 and a PhD in biology from Harvard University in 2008. I'm very pleased and proud to note that NIGMS has supported Trevor's work since 2014 and currently funds his research through a MIRA grant. And as if that wasn't enough, just a few weeks ago, Trevor was named a MacArthur Fellow as well as an HHMI investigator. So please help me welcome Trevor for his talk, Evolutionary Dynamics of SARS-CoV-2. Trevor, over to you.
Thank you so much, Dr. Lorsch, and thank you for the invitation, and it's great to be presenting to this audience. So for COVID at this moment, at least for the last 10 months, I've been really paying attention, kind of almost all of my focus has been on the variants of concern and variants of interest, alpha, beta, gamma, delta, et cetera. And so for this lecture, I'd really like to dive into what's happening here and look at the evolution of these variants and kind of what's happening now and what to expect in the future. So for the talk, I want to cover 7 different things, look at tracking virus evolution, emergence of these variants, current circulation patterns, adaptive evolution, transmission dynamics, genomic surveillance, and some on future perspectives. So first off, real-time tracking of SARS-CoV-2 evolution. Dr. Lorsch mentioned this in the intro, but one kind of key component of what we've been up to for the pandemic is running this nextstrain.org website. And so how this is basically working now is there's this database, GISAID, that people may be familiar with. That's where groups from all over the world are submitting sequence data. We pull data from GISAID. They're now up to 4.4 million genomes, which is remarkable. And every day we update the website, showing kind of what is circulating where. This has been a huge effort. We also provide tooling for public health groups and scientists throughout the world to use this open source tool set to look at their data from their locality or their sequences. And so in doing so, we've had kind of a view into the evolving virus since January 2020. So if we go back way to the beginning of the pandemic, originally this genomic data was most useful to understand just transmission patterns. So each mutation in the viral genome kind of gives you a label to see this lineage went here and this lineage went here and so forth. And along with the molecular clock allows us to build these time-stamped phylogenies, which are really useful for understanding what these patterns of spatial and epidemic spread. And so with early on, we're able to see emergence in Wuhan here back in December 2019, spread within China in January, February 2020, and then all of these introductions elsewhere in the world from China at this point. And then after this, then alongside this kind of original early period, there wasn't a lot of adaptive evolution or interesting evolution going on. The main thing to note is this D614G mutation. So this actually arises within China on these purple branches here. And then it's what seeds the epidemic into Europe here in this kind of green and yellow, which goes on to the US and so forth. And now basically all viruses descend from this D614G variant. We care about the spike protein mutation, D614G, in part because spike is the kind of critical protein for the virus. It's what actually binds to ACE2 on the human receptor and then spills the virus inside, where you can kind of make more virus and so forth. And it's also going to be the primary target for the human humoral immune response. So after this initial wave with D614G, we had this really heavy mitigation in spring and summer 2020. This results in all of these kind of individual lineages that aren't mixing so well. And so we have these clades or lineages that are largely disjoint throughout the world. And then we see kind of the first variants emerging at this point, but they're not so interesting. We have like a spike mutation just having 222V and a spike mutation having 477N. These early, due to this strong mitigation, these early variants don't really go very far. They're kind of confined to local regional dominance within Europe or within different geographies. But then we have the emergence of the variants of concern, which are kind of much better viruses, and they spread farther. So the main hallmark here was the spike mutations at 484 to K and 501Y. We started to see these pick up all across the world. We started to kind of detect them in December. They were occurring a bit before that. And then really interestingly, here this is, again, a phylogeny going up from the beginning of 2020 to March, April 2021. And you can see that, that there's this background of these kind of non-variant viruses, but all of these mutations again and again to at 501Y and 484K. You can see these really start to take off. And so those mutations correspond to the alpha, beta, gamma variants that you may be familiar with. So first, if we look at what happened here, if we look at alpha, or Pango lineage B.1.1.7, it emerges from the UK. It kind of appears to have all of these mutations that crop up at basically the same time in fall 2020, and then it rapidly displaces what was there before. These mutations include this really interesting 3 amino acid deletion in ORF1, ORF1AB, a number of mutations in spike, so 6970 deletion and 144 deletion, 501Y, 570D, 681H, and a complete knockout of this ORF8 gene, also interesting. And so again, we see that over the course of just a few months, this variant, the B117 or alpha, displaces existing diversity in the UK. We see a similar picture with beta, or Pango lineage B.1.351 in South Africa. This has the exact same 3 amino acid deletion in ORF1A. It has some of the overlapping mutations, particularly 501Y as alpha. And we see the same pattern where it rapidly displaces existing diversity. And if we look at gamma, our Pango lineage P.1, we see a similar picture. It emerges in Manaus in Brazil in late 2020. It has the exact same 3 amino acid deletion in ORF1A and some of the same mutations, including 484K and 501Y as beta. And again, we see over the course of months that this displaces standing diversity. So we have this pattern then that these variants of concern emerge. They have a bunch of additional mutations on top of what else is circulating, and it goes on to quickly spread and displace existing viruses. The working hypothesis that I have for the emergence of these lineages, emergence of these variants of concern, is that this is within host evolution, that we didn't see these occurring in the spring wave due to what happens, and you get this fall wave where there are people that were infected in the spring, and there's not so many of them, but there's enough that the virus is encountering people that are seropositive, and then within host pressure and those infections causes the virus to evolve. So some evidence of this comes from direct studies that we can do of within host evolution. These are largely done for persistent infections, largely in immunocompromised individuals. Just as one example, this was published in fall 2020, even before the variants of concern were noticed. This is a single immunocompromised individual who is sick with COVID for over 150 days. And then in that time, the virus within them evolves kind of more nucleotide changes than exists in a general lineage across the world, which is remarkable. But really importantly, if you look within this individual, we're getting some of these hallmark mutations that we were seeing in the variants of concern. So you see within this individual 51Y, we see 484K, we see the 144 deletion seen in alpha. And so this, to me, suggests that within host pressure is a mechanism that aligns with the mutations that we're seeing, the variants of concern. That within host pressure is I think, a pretty good hypothesis for what's actually causing the emergence of these variants of concern. So we look at current circulation patterns now. We can see that kind of all of these gray and non-variant viruses, we get the emergence of beta in South Africa, alpha in the UK, quickly spreading to Europe, gamma in Brazil, quickly spreading through the rest of South America. For a time period, there's kind of a soup of these different variants circulating, but then Delta emerges in India and then quickly displaces things throughout the rest of the world. So if we look here up until basically September, we can see this pattern where there was a time period in spring where there was a lot of co-circulation of these different variants. The Delta emerges from India and then very quickly goes to take over. So you can see that here in the phylogeny, where kind of Delta is quickly spreading as a clade. It is present almost everywhere in the world. It's been a bit slower to take over South America, but I think it will get there if we just give it a month, few more. And then if you look here at this kind of frequencies through time, we see at this point, or even in September, that Delta is basically almost all viruses globally. And this kind of time period from emergence in fall 2020 to taking over the world in just 12 months is remarkable, and is kind of that pace of evolution is really rarely seen. A new flu strain that emerges and like we need to update the vaccines to, that generally takes 3 or 5 years to take over the human population rather than just 1 year. Take over the virus population rather than just 1 year. Generally, one way that we look at these things, as we were just doing before, is to look at time on the X and the divergence on the Y, just a number of nucleotide differences. We can see that viruses that are circulating today basically have about 40 mutations different in them in the entire genome compared to the original viruses in Wuhan. You can see that in this time period where there's kind of co-circulation of the variant of concern viruses with the non-variant viruses, the variant of concern viruses will have generally additional mutations to them compared to non-variant viruses. And this is particularly acute if we look at the S1 domain of spike. So S1 should be where most of the humoral immune response is to. It's kind of what's actually sticking up on the surface. And we can see then that non-variant viruses having 0 or perhaps even 4 mutations in S1, whereas the variant viruses will have 8 or 10 or 12 mutations in S1. And so we have this kind of anecdotal picture here that much of the evolution of the variant viruses is focused in S1, but we really wanted to look at this in detail and to kind of look across the genome and see what regions of the virus are undergoing adaptive evolution. Okay, so this study was led by Katie Kistler in the lab. She's a PhD student, and with John Huddleston in the lab assisting. So here we built a phylogeny of 10,000 viruses that we attempted here to sample equitably in space and time. So we kind of look across countries and across months and try to take an even sampling of virus genomes. This is helpful in that certain countries like the UK are submitting a huge amount of sequences to the database, and we just looked at what's in the database. We're going to be biased towards the countries that are submitting lots of data. So this throws out a lot of data, which is unfortunate, but it helps to kind of remove bias. We've repeated the analysis with 20K and we get the same effect, but it would be nice to have additionally methods that work better for kind of very large phylogenies. This is generally an issue as the data starts to scale. Okay, so we build 10,000 phylogeny of 10,000 genomes sampled throughout space and time. And then one thing that the key thing that we then look at here is clade growth. So we can look at a particular point in time. So this is done in April 15th. And we look at clades that are circulating. So a clade is going to be a particular viral lineage that descends from a common ancestor. And there'll be clades that are growing in frequency that are kind of in red. This is actually delta here. And then clades that are declining in frequency here in blue. And we can infer that clades that are growing are going to be more fit by definition compared to clades that are shrinking in frequency. And so we can look at the clades that are growing and seeing what sort of characteristics does the virus have. And we can use those characteristics to then understand what is actually being selected for in the virus. So here, if we look at S1, non-synonymous changes, so amino acid changes, we can see on the x-axis, this is exactly what we're just looking at previously, where over time, we accumulate a number of mutations in S1, and that the variant viruses, as the colored circles rather than the gray circles, generally accumulate mutations more quickly. However, kind of the key result here is that if we look at viruses that have more mutations in S1, so these ones up here, they will generally be belong to clades that are growing in frequency compared to viruses that have fewer S1 non-synonymous mutations. So this correlation, Pearson correlation of 0.46, this is basically for the kind of PopGen molecular evolution folks, this is equivalent to McDonald-Kreitman test, and it's quite difficult to trick or to kind of get an artifactual result here, where if you have something where there's a significant correlation, I'd be willing to ascribe pretty strong belief that it's really the mutations in S1 that are driving clades to be more successful. So this tells us then that S1, that generally mutations to S1 are resulting in clades in viruses that are more fit and that are outcompeting viruses that don't have as many mutations in S1. We did a control analysis here to look at correlation in control regions to just check that the method is working as it should. So if we look at S1 synonymous mutations, things that don't change the amino acid, we can see there's fewer of these accumulate through time. But even if we look at that accumulation, where it's things that have more S1 synonymous mutations compared to logistic growth rate, we don't see a correlation. And if we look at polymerase and RDRP, this should be under strong functional constraint, and we also don't see a correlation here. So this suggests that this is likely a true result. We can look at other genes in the genome then and see where those how those compare to S1 in terms of their amount of adaptive evolution. S2 looks like it's doing something perhaps, maybe M, maybe ORF7A, but S1 looks like it's generally the focus. And we can compare everything here as this scatter plot where the y-axis is showing this correlation of how much selective advantage do mutations confer. S1 is at the top here, and then the x-axis is showing the evolutionary rate per codon, where S1 also stands out as higher in general. ORF7A and NSP6 are also significant. S2 is almost significant, but not quite. All of this stuff, which is generally kind of more functionally constrained genes as it should be, is evolving more slowly and doesn't show this clade growth correlation. ORF8 is this interesting outlier, which it tends to get knocked out repeatedly. So it looks like it's generally under less selective constraint. There might be some correlation here, but much of this fast rate of evolution is just due to the fact that you can knock out ORF8 and still have a virus that is functional and can replicate and transmit. Okay, so this would suggest that anecdotal observation that lots of the mutations and the variants of concern being in S1 is rigorously supported, and that S1 is generally the prime target for adaptive evolution in SARS-CoV-2 up to this point. We can measure this as well in this other kind of classic PopGen molecular evolution metric of DNDS. So DNDS is telling us the number of non-synonymous mutations per non-synonymous site relative to the number of synonymous mutations per synonymous site. So a DNDS of 1 would indicate the same rate of evolution in synonymous sites as non-synonymous sites, and would be consistent with complete neutrality, where like the gene doesn't matter. If DNDS is less than 1, this should indicate functional constraint, where generally mutations are purged by natural selection, and DNDS greater than 1 is consistent with strong positive selection. So if we look here across different genes in the SARS-CoV-2, we can see that initially, kind of corresponding to our anecdotal observation, there wasn't a lot going on in spring 2020. But as we go forward, this is taking the phylogeny and slicing it through time and looking at these slices and counting the number of synonymous and non-synonymous mutations in different genes. We can see that starting in fall 2020, S1 really picks up. And so at this point, S1 really stands out as having a DNDS of greater than 2. And maybe this will slow down at some point, but right now this is a really fast degree of adaptive evolution. To put that in perspective, if we look at H3N2 influenza, this is the fastest evolving subtype of flu. It's what we need to be updating our vaccines to every year or two. And this is now not in pandemic state, but in endemic state, but it's still H3, the hemagglutinin protein of H3N2 is held up as this is a prime example of ongoing selective sweeps, ongoing adaptive evolution. If we look at H3N2, HA1, the equivalent of S1, this has a DNDS of 0.4. So we see that at the moment, SARS-CoV-2 is adaptively evolving about 5 times the rate of H3N2 influenza. So again, it may slow down, but at the moment, the amount of adaptive evolution we're seeing is remarkable, is unexpected. And I don't think anyone would have expected this strong of DNDS back in spring 2020. So we can look at a couple additional things in terms of this adaptive evolution. So rather than just how much of it is, just more characterizing it. So here we see that if we look along particular lineages, that these mutations in S1 tend to cluster together. So rather than kind of having a regular tick of mutations, it's kind of tick, 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 and then it slows down. This is seen here if we look at wait times, where compared to our null expectation, there's a number of kind of wait times from one mutation to another that are very short. So this is consistent with either epistasis, where there are particular mutations that are kind of correspond to one another, or it could be due to particular transmission chains that are moving through these more seropositive populations compared to other transmission chains. There's kind of different levels of selective pressure across the phylogeny. So additionally here, if we look at, we can look to see convergent evolution. And this is a kind of another prime example of what we expect from adaptive evolution. We see the same mutation again and again. So if we look across this phylogeny of 10,000 viruses, we see a number of mutations that are occurring repeatedly, kind of more than we'd expect. These include 9 occurrences of 484K, 8 occurrences of this strange 3 base pair, 3 amino acid deletion in ORF1A, a couple other non-spike genes, but then 452R, which is in Delta, seen 5 different times, 95I, also in Delta, seen 5 different times, 501Y, seen 4 different times, etc. So just seeing these mutations again and again is indicative of convergent evolution and is consistent with adaptive evolution. And then some of these, whenever they occur, they generally cause a clade that is going to be more fit and to spread. And so these are particular highlights for individual mutations that are causing adaptive evolution. Again, we see this 3 amino acid deletion in NSP6. So there are particular mutations elsewhere in the genome besides S1 that appear to be adaptive, even if kind of per capita S1 appears to be the main focus for adaptive evolution. So generally then what we see here is that mutations in S1 are arising via within host pressures that result in an increase in viral fitness. And then after they've emerged, they're enriched in the virus population by natural selection because they transmit better. Each infected individual will cause more downstream infections compared to these non-variant viruses. And kind of maybe the bigger picture here is that even though we haven't seen very much antigenic drift up to this point, the original vaccine formulated against the Wuhan virus seems to work well against Delta, et cetera. This level of evolvability of the S1 domain suggests to me that once we start putting more selective pressure on the virus for antigenic drift, that it will be able to do so, and that we'll need to be updating the vaccines at a fairly regular basis as we go forward. And we should at least be prepared for that scenario. Okay, shifting gears a little bit. I want to talk here more about transmission dynamics and kind of looking in detail at one variant compared to another. So there's generally multiple approaches here to modeling fitness differences. So to kind of describe in big picture how this works, one popular approach is a multinomial logistic regression. And so this is looking at frequency through time. So blue here is Pango lineage B117, our alpha variant. Purple lineage here is Pango lineage B.1.617.2 or delta variant. And we can see that generally across different states, where in the US since around January, February, we have actually quite good genomic surveillance. We can see these waves of B117 or alpha that increase pretty rapidly, but then they all seem to be displaced by delta, where delta emerges, it quickly goes to 100% and pushes down other things. However, this doesn't capture the full picture in that there can be differences in caseloads compared to frequencies. And so maybe helpful here is that we have states like California here, which has a pretty big wave of alpha if we look at frequency data, but that didn't actually result in large caseloads. Whereas in some other states, those waves of alpha did result in epidemics like Colorado, Michigan, and Minnesota. But basically everywhere we've seen the wave of delta resulting in larger caseloads. And so here we wanted to be able to distinguish this and look rather than just at the fitness advantage, we wanted to be able to look at the actual RT, or the number of secondary infections that one infection causes of these different variant viruses. So this is work led by Marlon Figgins in the lab. Marlon is a PhD student in applied math. So this is looking in this model, it's kind of taking this observation and trying to formalize it and really put some rigorous numbers on this here. So what Marlon uses here is, as an example, this is Washington State, where we have as a model input the number of cases through time. So we have reported cases. This isn't going to be all infections, but it's going to be capturing a decent fraction of infections. And then we also have the variant frequencies as reported to GISAID, where we can see here is our alpha wave, our delta wave, et cetera. And so in the model, we use these state level case counts and we partition them based upon the frequency data to give us something like this, where we have then an actual estimated caseloads of the different variants. And then from these caseload trajectories through time, we can estimate RT, or the number of secondary infections caused by one infection, where we can see in this case that delta starts off at 1.8 secondary infections and then drops over time to perhaps 1.2 at the by the end of August. And we can then compare these RT values to look at the different growth advantages of the variants. So when we do this across states, we get this pretty interesting picture here. This is showing non-variant viruses compared to alpha, beta, et cetera. And then each line here is the trajectory for a particular state. We have 46 lines here for states that have enough data. And we can see that even in March 2021, non-variant viruses were all dying out. They have less than, an RT less than 1. And if variant viruses hadn't evolved, hadn't emerged, the epidemic in the US would have been over by May, June, July. However, different variant viruses have increased transmissibility and then cause RT to be above 1 where non-variant viruses aren't. So alpha is still spreading and still increasing in frequency up until April, then it declines below 1. Same story for beta, same story for gamma. Mu appears to be a bit more transmissible, but still not nearly where delta is. So we look at delta, its RT back in May is around 1.5 or so, and it's still above 1 at the beginning of September, and it only starts to drop below 1 in October. We can then compare across these different variants to look at what the relative transmission advantage is. Each dot here is a single state. There's going to be 46 dots here, 46 independent estimates of alpha. We run the model completely separately on each of these different states. And so having consistent estimates for each state gives us kind of some reliability in the model or some belief in the model's outputs. Where if we look here at alpha, we see that this has a transmission advantage relative to non-variant viruses of perhaps 1.3 or so, a little less for beta, a little more for gamma, a bit more for mu at a perhaps 1.6 or so, but really delta is the standout here, where it's about 2.2 or so times more transmissible than non-variant viruses. So again, kind of this level of evolution where we increase fitness by 2.2-fold in just a handful of mutations is remarkable and unexpected. And this delta increase in transmissibility of delta then results in this large wave in the US and what we've seen with large waves elsewhere, surges elsewhere in the world. However, interestingly, there is some difference in the titers, in the neutralization titers across these variants, with beta, gamma, and mu having more of a drop in neutralization titer relative to alpha and delta. It's not a huge difference. What we see if we look at this model, we can compare the state-specific and time-specific RT values for each of these different variants, and we can compare them to the amount of vaccination that existed at that state at a particular time. And we can estimate how much is vaccination actually reducing RT, which is of course very important. And interestingly, we see similar reductions in RT for alpha, beta, gamma, delta, and mu. And so it's just that delta has increased things to such a degree that even though vaccination works well against it, it still hasn't been able to avert this, this wave just due to how transmissible it is, even though most of the infections are happening in the non-vaccinated population. So, so I'm really happy with this general framework of having case counts and partition frequencies. And this really lets us understand fitness in a very rigorous fashion. And this is something that kind of I would have, like as a population geneticist, it's really amazing to have this data of very rich frequencies through time. It's kind of exactly the data you'd want to estimate fitness. But now maybe the next step of what we're trying to do is how do we tie this to particular mutations or phenotypes, neutralization titer, et cetera, to be able to understand this more kind of linkage from actual genotype to fitness. One study here that was just published to medRxiv, this is Fritz Obermeier and colleagues at the Broad. They do this really interesting model here where they do a logistic regression model, and then each mutation will have a particular effect. And so the fitness of a pango lineage, the fitness of a virus, is the additive effect of all of these individual mutations. So when this is run on circulating viruses, we see that delta is more fit, has a higher fitness than almost anything else. Mu is a close second. Other things are less so. This mirrors the results that Marlon was getting as well. We see that at this moment, there's not a lot of variation within delta. There's kind of these two main lineages, which we at Next Strain have called 21J and 21I. There's some additional ORF1A mutations here in 21J. That's increased things a little bit, but not hugely. So right now, there doesn't seem to be a lot of fitness variation within delta, and we're basically waiting for more mutations to emerge. What's been interesting here and surprising, and I'm not exactly sure, maybe a bit mysterious, I'm not exactly sure how to explain, is that we saw in all of the other variants, again and again, we had mutations mutations at 44K and 501Y. And in each of those cases, they were seeming to increase the fitness of the virus and causing those variants to spread. However, in Delta, we've basically not seen this. And it's not because it hasn't happened. Like we can easily find 44K and 501Y viruses within Delta, but those aren't seeming to take off. My best explanation here is that there's an actual epistatic effect and that these mutations are not as beneficial on the Delta background, but this needs more study. It's certainly important. However, what we are seeing at the moment is even though there's not some kind of major things occurring within Delta, we're starting to see local sublineages that appear to be perhaps a bit more fit. So as just one example, we have AY.4.2. This has spike mutations 145H and 222V within Delta. It's been spreading within the UK. So here it's in blue here. It's about 10% frequency now, arising from about 1% in August. This corresponds to about a 10% fitness advantage. And we're seeing kind of similar stories elsewhere. And so what I suspect will happen is over the following months, unless there's kind of another big innovation, which I guess we could see, that we'll have kind of more and more of these local variants that start to spread a bit, and there'll be kind of competition of things within Delta, but it's not kind of one thing taking over the same way that we had before. But of course, yeah, something could well emerge. Okay, so to watch for this occurring and the emergence of these variant of concerns, we need genomic surveillance. This has actually been a remarkable achievement of the global scientific endeavor during the pandemic. So if we look in GISAID and we look at August 2021, just as an example, there's 630,000 genomes shared. Just 4 years ago, 2,000 genomes for an Ebola epidemic was a huge number. And if we look over time, as specimens are collected, they're generally sequenced and submitted shortly after collection. There's some things that are more backlogged, but by and large, the genomic surveillance is having great turnaround times. And so the number that I really like to think about then is be able to have eyes on things. What matters is having kind of genomes available today for the last month. So I like to look at is the number of sequences available at a particular point in time that were collected, sequenced, and shared in the past 30 days. So if we look globally, this hit a peak of about 250,000 genomes available from the previous 30 days in September, has declined a bit, but generally this is hugely remarkable. You can tell that people kind of got in on this and really started to think this was really important to sequence and share quickly once the variant of concern viruses started to pop up in December, January, and then everyone starts to sequence more and invest in it. However, this genomic surveillance is not completely even across the world, as you might expect. If we look in the US, UK, and Europe minus UK, we're all doing fairly equivalently of 10, 20,000, 80,000, 100,000 genomes shared in the last 30 days. This gives a huge window into what's circulating. This allows the methods like what I was presenting from Marlin to work, where we have detailed frequencies through time. However, if we look at, say, Africa and South America, they've really picked up dramatically, and I'm not nearly really worried about on the kind of continental level of missing variants of concern. There are particular countries in Africa that still need better genomic surveillance, but this is starting to look much better. But still, if I could kind of wave a magic wand and have an additional, 10,000 viruses sequenced per month, it'd be so much better to have them come from South America, Africa, than just more coming from US, UK, Europe, where so much sequencing is already occurring. So generally, genomic surveillance should be aimed at rapid turnaround times. And then what's really also important is being able to have this tied to data systems, so we can know that this sequence resulted in a hospitalization or a death. This sequence came from was a breakthrough infection and a vaccinated individual, et cetera. And having those data systems to tie things together is something that the US has been falling behind on relative to, say, the UK, where being able to link that data is really important to look at secondary attack rates and vaccine effectiveness across variants and so forth. It's something that we should really be investing in more. Okay, so in the last 5 or 10 minutes, want to touch on some very broad future perspectives from this kind of evolution that we've seen to date. So part of this is maybe a bit of bias in coming from flu, where I was been working in seasonal flu for over 10 years now. And so it makes it kind of an easy way for me to do an analogy by comparing SARS-CoV-2 to influenza to try to understand what it will look like in endemicity. So for each of our flu pandemics, it's a similar situation where there's a pandemic in 1968 or 2009. A lot of people get sick with flu, but then after the pandemic, it goes back to kind of an equilibrium point where year after year, the virus is evolving, and then it's infecting people in this endemic state. And people are still getting sick, people are still dying, but it's not at pandemic levels. And so I want to compare to get an idea of what endemicity might look like in SARS-CoV-2. We can compare to influenza. So the 3 main things that I'd look at here is the R0. So this is the number of secondary infections in a completely naive population. R0 of Delta is perhaps 5 or 6 at this point. So the original Wuhan-like virus was 3. Delta is perhaps 2 2 times this. And then R naught of flu is perhaps 2. So even if everything else was the same in terms of evolution and waning immunity and so forth, we'd expect more circulation of COVID than seasonal influenza just because R naught is higher. So far, at this moment, we have rates of adaptive evolution S1 that are about 5 times faster in SARS-CoV-2 than H3N2 influenza. I'd expect this to slow down as the virus kind of loses out, captures these easy wins and they don't exist for it anymore. But this level suggests that S1 is going to be quite adaptable, and I would expect some degree of antigenic drift. We also know that there's a decent amount of waning immunity from looking at SARS-CoV-2 vaccine studies, as well as looking at seasonal coronaviruses. And then a pretty big question here, but I would guess that IFR of COVID is roughly comparable to flu once you have prior immunity. So we might have tenfold less hospitalizations and deaths per case in the endemic state than in the pandemic state. And so if we put these three together, we get a virus that circulates like flu does, perhaps higher levels given R naught. Individual infections aren't so bad, but there are a lot of infections that are happening every year. And so if we do out some very rough math here, we'd expect in the US perhaps 50,000 or maybe 100,000 deaths in the US each year. This is like flu times 2 or 3, basically. And so something that is going to be an important thing to be addressing in the coming years. So it's not the cancer or heart disease, but 100,000 deaths or 50,000 deaths is a lot that we'll have to deal with. And the thoughts here and kind of strategies are that kind of on the shorter term, if we can do a good job forecasting evolution, the mRNA vaccines give us a really ability to do fast turnarounds. So with influenza vaccine, we need to select strains in February because it takes long to grow it up in chicken eggs and then distribute. But instead, if we could have maybe a decision in June or something for a fall vaccine campaign for these annual booster shots, we could have probably a pretty good match to circulating variants. We could also imagine putting in multiple valencies into these mRNA vaccines. Again, trying for as good of a match as possible of what's in the vaccine to what's circulating. On a slightly longer time scale, if we could figure out some of these universal vaccine strategies to either get a broader immune response or target more conserved epitopes, this should work better in terms of vaccination. I do think that there's still a role for testing, contact tracing, public health apparatus to combat things, and that if we can strengthen this, it will help with our seasonal ability to respond to seasonal flu and COVID outbreaks. But if there's another pandemic or some novel pathogen, it also is the kind of appropriate infrastructure to deal with that as well. So it wouldn't be a lost cause. And then we also have more kind of obvious things like improved treatments. I'm really hopeful for these new antivirals, but also just improving ventilation, which could do a whole lot. And so it's going to be an ongoing public health burden, but something that we can continue to develop tools to address as time goes on. And so with that, I'm going to stop. I'd like to thank data producers from all over the world. This sharing of sequence data has been absolutely remarkable. GISAID for their amazing curation, NextStrain team, and also particularly Katie, John, and Marlon for their work that I was talking about today. And happy to answer your questions at this point.
Thank you so much, Trevor, for an extremely enlightening talk and a very, very important series of studies. I've been asked to just repeat the CME code. It's 37917, 37917. We have a lot of questions, so I'm not going to be able to get to them all. I'm going to have to sort of pick ones that I think maybe you didn't touch on immediately and would be worth discussing. So this is one from Kayam, who asks, are the deletions in ORF1AB due to common ancestry or convergent evolution?
Yeah, so it's a good question. There's been questions about recombination, which should start to pick up. What we've seen for these ORF1AB and SP6 deletions is there are definitely convergent evolution that's occurring in all these different lineages, which is especially remarkable. And like I can imagine it's much easier to get convergent evolution for a single nucleotide change, but getting a 3, kind of the alignment of this 9 base pair 3 amino acid deletion again suggests that it's a very kind of important fitness effect. We have evidence from kind of in vitro virological studies that NSP6 modulates the interferon response. So you might think that there's some interesting function there. I haven't seen anyone actually do that in vitro study of the NSP6 deletion versus the non-NSP6 deletion to kind of dig into what it's doing, but I suspect it's something pretty interesting given that we see it again and again.
Interesting. So this, let's see. And another question from actually the same person, Kyle. Why does DNDS of S1 seem to start changing faster in late 2020?
Yeah, this is a bit mysterious. The hypothesis is that in late 2020 is when we're getting this basic second waves in much of the world. So we had spring waves in much of the world, US, Europe, et cetera. And then we have fall waves in autumn 2020. And then at those waves, a certain amount of individuals that the virus is encountering will now have been infected before. Some of those individuals, through their natural immunity, will be completely have sterilized immunity. They won't get infected. Other individuals will have had lower titers. They get infected, and then within that person, there's now more selective pressure for the virus to evolve to escape immunity by getting better at binding to ACE2 in the case of like 51Y, but also better at escaping their antibodies in the case of 484K. And so, yeah, the hypothesis is that then it's this fraction of infections in seropositive individuals that that are escaping immunity in some fashion, and that those are what is increasing DNDS. Whether that, how exactly to determine that it's really within host pressure is a little tricky, but that would definitely be the hypothesis.
Okay, great. This may be related to that answer actually, but Debananda wants to know about studying the effects of the vaccination. So, once vaccination took place, obviously immunity increased, and were you able to get information about how that affected the generation of variants that could then cause resistance to the vaccine?
Yeah, this is a good question. And I remember in giving kind of talks on influenza, I'd always get this question as well of like, how much is the vaccination affecting evolution of the virus? For a couple things here. So one is that the fastest rate of evolution will be when there's lots of circulation, just generating mutations and so forth, and lots of selective pressure. So if you have vaccination, it will kind of be at odds between these two things. We'll increase selective pressure, but we'll decrease circulation. And so overall, whether that increases evolution or not is maybe unclear, but it's going to be less compared to the scenario where you have an epidemic that increases infections as well as immunizing people during that epidemic. What we've tried to do here, and I don't have public data on this yet, but I hope to have that soon, is by taking that same analysis that I presented from Katie. And so what Katie's done is taking this tree of 20,000 tips, slightly larger tree, and then we can look on each node in the tree and see where do we infer it to exist via phylogeography, and when do we infer that node to exist? Does it have S1 mutations? And what was the vaccination coverage of that country at that time? And we can actually look across the tree to see, is there a correlation of vaccination coverage and rate of S1 evolution? And we don't see any correlation there. So there might be some effect, but we're not picking it up, and we're not seeing an effect of vaccination driving evolution in that analysis.
That's at least somewhat encouraging, I guess. Akshay has a question. Is there a maximal level of viral fitness that SARS-CoV-2 can achieve, or will it continue to evolve to be ever more transmissible?
Yeah, so there's like a meme of viruses evolve to be less severe over time. I don't think we can really rely on that here, in that it's not killing enough people to cause a lot of force of evolution. But what should happen is basically the virus wants to have as high of kind of viral load as possible to be able to transmit more. But if you keep increasing viral load, you're more and more likely to put someone sick in bed. And so there's going to be this trade-off that if you increase your kinetics and have viral load spike even more, you have someone staying home when they're sick rather than out in the community. And so that's going to, at some point, that's going to limit the R naught of the virus. But whether it kind of can go from 6-ish now to 8 or 10 or whatever that kind of ceiling actually is not entirely clear.
So Roland would like to know, do you think the rapid adaptive evolution of SARS-CoV-2 versus the flu could be a function of its recent jump to humans versus the flu, which is?
Yeah, I think certainly. And much of that additional DNDS is not due to antigenic evolution. So the 0.4 DNDS number I had for H3 and 2, flu is all antigenic evolution, and it's kind of escaping from immunity over time, whereas SARS-CoV-2 is all becoming better adaptive to binding and replicating and so forth, which should slow down. So much of that high rate is definitely due to it being a pandemic virus. But what's been weird is that, like, even if we look at 2009 flu pandemic, it infected a third of the world in the course of 2009 to early 2010, but there wasn't that big jump in DNDS of flu at that point. There was like a slight increase. So yeah, I don't know exactly what to take from that, other than, yeah, again, that SARS-CoV-2 is quite adaptable.
So I guess on the other side of that coin, Cecile has a question, again, comparing flu to SARS-CoV-2. The evolution of the spike protein in the coronavirus is much faster than flu H3N2 HA. Yet there's mounting evidence of cross-protection between SARS-CoV-2 lineages. And is immunity to SARS-CoV-2 just better across the board so that antigenic drift may not be as much of an issue relative to the pace of evolution? Is there a good understanding of the mapping between evolution, immunity, and clinical protection across these kind of RNA viruses?
Yeah, it's a big question. So for flu, the way that we often think about this is in terms of drop in neutralization titer or drop in HI titer in the case of flu, but it should be pretty similar. And the WHO has their bar for updating the vaccine when there's an 8-fold drop in titer. Delta was a 6-fold drop in titer. Beta is about a 10-fold drop in titer. And we're only whatever 20 months in at this point. If we look at 2009 swine flu, that took a good 3 years or so to start to see any antigenic evolution, in part because that first round is just getting to all the naive individuals. So up to this point, we've seen what would be considered kind of some antigenic variants in flu that we would be updating the vaccine to if we were just making this like we make flu decisions. But they haven't been huge changes. Again, yeah, about 6-fold, 10-fold in the course of these 20 months. That is about where H3 and 2 is, that we get about this, we get about 4-fold, 8-fold every year or so. So it's not, so far it doesn't seem too different. It's just perhaps been less time in comparison.
Interesting. So here's a conceptually simple, seemingly simple, but conceptually probably very important question from Tom. So how can a virus strain push down another strain? You know, you saw the curves, one goes up and then another one seems to push it down. What's the mechanism behind that kind of takeover?
Yeah. So yeah, deceptively difficult question. So the actual mechanism is almost certainly going to be that there's alpha spreading and beta and delta spreading. And if delta gets to someone first, they recover and alpha can no longer get to them. And so there's an actual interference there. But why this is actually difficult is we still think that there's a lot of susceptible individuals in the entire population, that you can imagine kind of an epidemic of alpha happening at the same time as an epidemic of delta, and why don't they just stack on top of each other rather than kind of interfering? And so the degree of actual interference, because it's not completely zero sum, rather than like the classic PopGen models where everything is zero sum, is it's super tricky to figure out exactly what's going on there. We think there has to be interference. Otherwise, mu and what we were just presenting, mu has a fitness advantage. It's quite good. It should have been able to take off in the US, but Delta got here first, spread through enough of the population that it immunized enough people that it kept mu out. So it's certainly this immunological cross protection, but the degree at which one infection is actually interfering with other infections, whether that's 100% or 0% or where in between, is difficult to actually infer from models.
You think there's an intersection of behavior, human behavior with that? So I mean, there's a pool of people who can be infected, but maybe there is a pool that is more infectable at a given time, and that's how it gets in. Is that something that's modeled?
That's definitely fair. This fits with, I haven't worked on this at all. This fits with this network modeling where you can have kind of hubs in these social networks that kind of cause more spread than other individuals. As you'd imagine, these alpha percolating and delta percolating, and the first one to get to that hub will then kind of have a greater effect at blocking onward transmission. And if those hubs are people that are kind of out and about and so forth, behaving differently, you might expect that to play a role.
So that's very interesting. So Amelia wants to know, do the observed convergent evolution mutations cause the SARS virus to converge on, I guess, the phenotype of the common human cold coronaviruses, phenotypically or antigenically or in other measurable ways? So is it going to be just like a common cold, or is it going to stay with the death rate that it has?
I think the big difference here is not the evolution of the virus, but much more about prior immunity. So the reason that the endemic seasonal coronaviruses largely do not make people so sick is because even as they evolve, everyone was infected as a child, and again as they've throughout their adult lives. So each time they encounter the virus, there's some immune memory. And so there will be some degree of kind of convergence in endemicity at the endemic state between seasonal coronaviruses and SARS-CoV-2. But I think that is largely explained by immunity rather than by actual evolution towards the same place.
So Celine has a question about a new variant. So what do you think about the new Delta variant AY.4 with SY145H mutation of the spike? Is this known or predictable that this variant's more transmissible?
Yeah, I had a slide on this. So we see 222V in particular, which that also has, that like came up independently in fall 2020, and it's something that we think should have an advantage just by itself. I think there was less suspected on 145 by itself. So it appears to have a slight transmission advantage if you just run the numbers, which I pretty much trust how those work at this point, given that we've seen them work pretty well in the past. We get this 10% or so fitness advantage. Whether that will, like how far that will spread when other things are emerging to compete with that 22V 145H is not entirely clear. So it's not, that in particular wouldn't concern me. Just the general picture of how easily SARS-CoV-2 is able to evolve and that these things will keep appearing is much more of a concern.
I think we have time for one more question, and Akshay has an interesting question. So you mentioned the importance of continued contact tracing potentially. So Akshay is asking about, is it going to be possible either soon or in the more distant future to combine contact tracing with the kind of genetic epidemiology you're talking about, to actually say that this person gave that person the infection based on looking at the sequences?
Yeah, this would be where things should be headed, certainly. What's made that difficult for much of the kind of academic researchers is you need that very identified data that is really hard to work with. Where there's been some interesting success here is in state health departments, sort of public health entities, actually pulling in the genomic data into their case investigation or their contact tracing investigation. And we've been like we've been trying to help with that with the NextStrain tooling. And there's some kind of interesting stories that have come out of that work. And there's a number of kind of one-off papers that look at these very detailed studies of spatial spread. New Zealand is doing an amazing job with this, for example, where they're actually sequencing every single case that they have low enough CT for. So that comes in at about, they're getting maybe 60% of things sequenced, and they have the full interview data and everything, and they're able to often rule in or rule out particular epi links with the genomic data. And it's and that works in part because there's a few hundred cases or that sort of level that you can reason with it. But once you have like US levels, that just gets really, really difficult. But if you're in this sort of containment regime, it's super helpful.
Well, with that, I'm afraid we have to end. We still had quite a lot of questions. That shows the enthusiasm for the work you're doing. I want to thank you again for this talk, for the work you're doing, and say how pleased we are to be able to support you now and in the future. So thank you so much, Trevor, and thank you everyone for watching.